Hello everyone, welcome back to study session. Today we are going to be discussing about prokaryotes. So let's get started. Prokaryotes. It is essentially bacteria. So bacteria are unicellular organisms and they are actually prokaryotes. Now in a previous video I have discussed about cells and the two main types of cells, that is prokaryotic cell and eukaryotic cell. Please go check that video out if you want to learn more about eukaryotic cell and the differences between prokaryotic and eukaryotic cell. In this video we are going to be only focusing on prokaryotic cells or bacterial cells. So prokaryotic cells can be classified into two different groups. First, Archaebacteria and then eubacteria. So what is Archaebacteria? Archaebacteria are the bacteria that can live in very extreme environments like hot springs, volcanic lakes, lava muds. So extreme temperatures, extreme pH, Archaebacteria can live in. Now this might be unusual in today's date, but it was actually quite common in primitive Earth because primitive Earth, after all, had extreme environments and Archaebacteria were the one that actually could survive in those environments. Now, eubacteria is commonly what we see in a very wide range of environments, including soil, water, and in human as pathogens mostly. So let's discuss a bit more in detail about a certain type of Archaebacteria, and that is thermoacidophiles. So thermoacidophiles, they can be found in hot sulfur springs. So very extreme environment for a bacteria, but they are genetically able to survive in that environment. And we are going to be also discussing about temperature conditions in which they can survive. For example, they can survive in temperatures up to 80 degrees centigrade and a pH as low as 2. Now 2 is quite an acidic pH for bacteria to survive in. However, thermoacidophiles can survive at a pH range as low as 2. Now bacteria. Bacteria in general, they are unicellular organisms. They don't have any membrane-bound organelles. They have a single circular DNA. And the majority of bacteria actually secrete a cell wall outside their cell membrane, which is generally composed of peptidoglycan or mucopeptide, muramic acid. Most bacterial cells would be spherical, rod-shaped or spiral with diameters of 1 to 10 micrometers. Now spherical bacteria, the shape, according to their shape, they are known as coccus for singular and cocci for plural, and similarly rod-shaped or spiral can be bacillus or vibrio. Now their DNA contents, they can range from about 0.6 million to 5 million base pairs, an amount that is sufficient to encode about 5,000 different proteins. Largest and most complex prokaryotes are cyanobacteria, in which photosynthesis actually evolved. So next, bacterial cells can be classified according to their ability to retain Gram stain or crystal violet after washing with a suitable organic solvent. So what is this? So basically Gram stain is a type of stain. So you can stain bacteria with this Gram stain, and if they can retain the stain, then they will be Gram positive, and if they can't retain the stain, then they will be Gram negative bacteria. Now the main differences between them would be Gram-positive bacteria may have up to 95% of mucopeptides, whereas Gram-negative bacteria may have as little as 5% mucopeptides. Gram-positive bacteria, they contain like peptidoglycan and teichoic acids in their cell walls, whereas Gram-negative bacteria, they contain substantial amounts of phospholipid and lipopolysaccharide that anchor proteins and carbohydrates of the surrounding capsule. Now, Gram positive bacteria will retain a crystal violet color upon staining, as you can see in this microscopic picture. So the picture, this is how gram-positive bacteria would look under a microscope after being stained by gram stain. So as you can see, they are clearly violet in color, and which means they have retained the gram stain, and by this you can tell whether they are gram-positive or gram-negative. Now, similarly for the gram-negative bacteria, you can see in this microscope image right here that the bacterial colonies, they look pink. So they, if you compare it with the gram-positive one, under the microscope it is clearly violet, whereas gram-negative is pink. So you'll find them to be pink in color. That is naturally the safranin. So they are dyed first in safranin and then in gram stain. So this is naturally the color of the safranin that can only be seen in a gram-negative bacteria, and you can't see the violet, crystal violet color. So that is one way of distinguishing between these two types of bacteria depending on staining. And then some examples of gram-positive bacteria would be Micrococcus species and Streptococcus species, whereas some examples of gram-negative bacteria would be, well, E. coli, because most of the gram-negative bacteria are pathogens. Now we are going to do a study on a specific bacteria here. Here it is E. coli. So E. coli, it is a very common bacteria that is found in human intestinal tract. So in the human intestinal tract, you can find a thriving colony of E. coli. And so, like I said, they are common inhabitants of the human intestinal tract. Now, these E. coli would naturally be rod-shaped. Now, they are gram-negative bacteria as discussed in the last slide, though, so they won't stain with the gram stain. And they are under eubacteria class, and they can be about 1 micrometers in diameter and about 2 micrometers long. They're surrounded by a rigid cell wall, which is composed of polysaccharides and peptides. Within the cell wall is the plasma membrane, which is a layer of phospholipids and associated proteins. Now, the DNA of E. coli, it is a single circular molecule in the nucleoid, which in contrast to eukaryotes is not surrounded by a membrane. So in eukaryotes, we have discussed it in the video of eukaryotes that they have a linear chromosome, and this is in the nucleoid, which is in the nucleus, that is membrane bound. But however, the DNA in the E. coli is not in a membrane, and it is a circular molecule. Now, the cytoplasm contains approximately 30,000 ribosomes, which account for the granular appearance that we see in an E. coli. Now, next topic would be bacterial conjugation. Now, bacterial conjugation is a pretty complicated and complex topic. However, we are going to do an overview and touch over the subject here. So, as you can see in the animation of the bacteria, two bacteria that are undergoing conjugation, first of all, some bacteria, they can carry filamentous appendages called pili or pili. Now, they can be usually like less than 10 nanometers thick and less than 1 micrometers long, and they will be attached to the surface of the bacterial cell wall or capsule. Now, here you can see there's a donor bacterial cell, and that is the one on the left. So the bacterial cell on the left is the donor bacterial cell, and it has this pili or the appendage that comes out from its cell wall or capsule. And what it does is that the donor is attaching to the recipient cell, which is on the right, through the pilus. So the pilus or pilus is actually helping the two bacterial cells to attach with each other. Now what happens is that inside the donor cell, which is the cell on the left, you can see there's a extra pink circular bit along with the blue one, which the recipient cell doesn't have here. So this pink bit is actually an F plus plasmid. Now what is a plasmid? A plasmid is actually a circular piece of DNA. It is smaller than the chromosomal DNA and it actually replicates independently of the chromosomal DNA. Now this plasmid here carries genetic information. Obviously what it is doing here, it is passing this information to the recipient cell via the plasmid through the pilus. So what is happening here is that this plasmid is again, it has double-stranded DNA in it, right? And this strands, the two strands, they separate. And what happens is that one strand of the plasmid goes through the pilus into the bacteria, into the recipient bacteria. As you can see in the animation, one strand is going through the pilus into the recipient bacteria. And what happens in there is that the recipient bacteria actually forms a complementary strand to the strand that has just accepted. It forms a complementary strand to it and hence forms a full complete plasmid in here, that is in pink. Whereas same thing is done by the donor cell as well, because now it is single stranded as well. It also forms a complementary plasmid and returns to its F plus plasmid state. So now both of them are in F plus plasmid state and they both are sharing some similar genetical information. So it is kind of a way of passing down information, even though bacteria, they have asexual reproduction. In this way, it is a way of transferring bacterial information through conjugation. So what is a plasmid? The definition is that a plasmid is a small circular piece of DNA which replicates independently of the chromosomal DNA. Now next we are going to discuss about bacterial flagellum. Now bacterial flagellum, here is two diagrams of the flagellum, and we are going to discuss about the different parts soon. So bacterial flagella are helical. They are 20 nanometers thick and several micrometers long. Now if you see the picture on the left, you can see the flagellum. The helical structure you can clearly see there, and you can see that it is formed of small subunits, and these are actually subunits of protein, and this protein is called flagellin protein. Now flagellum here, it is connected at one end and it is free at the other. So the free end is motile. Now if you see at the picture B, the figure B or the picture on the right, the white base region of a flagellum here is called a hook. And this hook, it actually connects the filament to the motor portion of the flagellum called the basal body. So as you can see, the basal body is the part that is anchored to the cytoplasmic membrane. As you can see here, it says outer membrane, peptidoglycan. So this is the part that is inside the cell basically. Now, as there are presence of rings, there is L, P, S, M and C rings, and these are surrounded by pairs of MOTA and MOTB proteins. Now these proteins, their main function is actually to drive the flagellar motor, causing the filament to rotate in a circular motion. So it's like for the rotation of the filament. But then you can see another set of proteins called FLI proteins. So here you can see near the C ring FLIG, FLIN and FLIM. So what they do is they actually function as the motor switch and they reverse the rotation of flagella. And this reverse of rotation of flagella happens if an intracellular signal comes through. So the MOT proteins, they have the function of driving the flagellar motor, causing the rotation of filament, whereas the FLI proteins, they actually function as motor switch and reverse the rotation of flagella, which happens in response to intracellular signals. Now, types of bacteria based on arrangement and location of flagella. So there can be 5 types of bacteria depending on the flagellar location and arrangement. Now the first type is Atricus. Now Atricus bacteria, they actually do not have any flagella. So these are like without any flagella. The next type is Monotricous bacteria. Now these have one single polar flagellum. So flagella at one end, one single flagella at one end. Here in the picture you can see a representation of the Vibrio cholerae with a single flagellum at one end. Now the next type is Amphitricus. Now these are the ones which have a flagella at either end. So at both ends they would have a flagella. And for example, there is Alcaligenes faecalis. And the next type is the Peritricus bacteria. So what they do is they have flagella all over their body and throughout, so surrounding the bacterial cell. For example, Salmonella typhi and E. coli. And then the last type is Lophotricus. Now Lophotricus is the bacteria that have like a tuft of flagella or a cluster of flagella at either one ends or both ends. In the picture shown here, you can see there is a tuft of flagella or a cluster of flagella coming out from one end, and this is an example of a Spirilla species. So 5 types are Atricus, Monotricus, Amphitricus, Peritricus, and Lophotricus. So let's discuss the key points from the lesson today. And the first point would be that prokaryotes can be classified into Archaebacteria and eubacteria. Archaebacteria, they can live in extreme environments like high temperature, low pH. I mean, if you say about conditions, but environments would be hot springs, volcanic lakes, lava muds. Prokaryotes are bacteria which are single-celled organisms with no membrane-bound organelles and single circular DNA. Bacterial cell walls are usually composed of peptidoglycan. Bacteria can be classified into gram-positive and gram-negative depending on their ability to stain violet with gram stain. So if they can stain violet with gram stain, they are gram-positive. If they can't, then they are gram-negative. Bacterial conjugation is a process of transfer of genetic material via plasmid through pilus. Now bacteria can be Atricus, Monotricus, Amphitricus, Peritricus, or Lophotricus depending on arrangement and location of flagella. So that would be all for today. If you like my video, please give it a big thumbs up and subscribe for more. Thank you.